Good day. Today our lecture about causative agent cholera disease, about Vibrio cholera. Cholera is life-threatening secretory diarrhea quarantine disease caused by Vibrio cholera serogroups O1 and O139, characterized by induced by enterotoxins secreted of Vibrio cholera. Vibrio cholera was first isolated by Koch. The old name of Vibrio cholera, Vibrio coma, in 1883 years, from cholera patients in Egypt. Though it has been observed early by Pattini in Italy. Taxonomy: domain bacteria, phylum Proteobacteria, class Gamma Proteobacteria, order Vibrionales. For us, important information about family Vibrionacea, genus Vibrio, species Vibrio cholera, which cause cholera disease. And genus of Vibrio includes another species of Vibrio, Vibrio parhemolyticus, Vibrio vinificus, minicus, and alginolyticus. For us interesting, toxigenicity Vibrio cholera and classified by two serogroups, O1 and O139 serogroups. O1 serogroups classified by two biotype, classical and L-Tor biotype of Vibrio cholera. I will explain about it, why this classification is present. And O1 serogroups classified by Inaba, Ogawa, and Gi-Koshima serotypes by A, B, and C antigens. Morphology: Vibrio cholera gram-negative microorganism, curved or comma-shaped rods with a single polar flagellum. Vibrios are highly motile. In stained film of fresh mucus from acute cholera case, the appearance as fish in stream. This type of motility is called as darting motility and suggests a swarming of gnats. Cultural characteristics: the cholera Vibrio is aerobic and slow growth in anaerobic condition. It grows within a temperature range of 16 to 14 degrees, optimal cultivation 37 degrees. Growth is better in an alkaline medium, it's very important, and it occurs freely between pH 7.4 and 9.6. Optimum pH for cultivation of Vibrio cholera 8.2. Vibrio cholera is a non-halophilic Vibrio. Vibrio cholera grows without salt. Most other Vibrios are halophilic. It grows well on ordinary nutrient media. Ordinary nutrient media for cultivation of Vibrio cholera: nutrient agar. Colonies are moist, translucent, round disc about 1 to 2 mm in diameter with a bluish tint and transmit light. You can see on pictures this petri dish with colony of cholera, Vibrio cholera. On McConkey agar, the colonies are small than those of nutrient agar and are colorless, but become reddish on prolonged incubation due to the late fermentation of lactose. On this slide you see colony of Vibrio cholera. On surface McConkey agar, lactose negative, colorless colony. Maybe after a long period of incubation, colony will have slightly pink characteristics. Also, blood agar. On surface blood agar, colonies of Vibrio cholera are initially surrounded by a zone of greening, which later becomes clear due to hemolysis. Aerobic incubation of the plate for no longer than 20 to 24 hours, also not optimal for detection of hemolysis, will permit differential of strongly hemolytic strains. In gelatin stub culture, liquefaction of gelatin begins at the top, which spreads downwards like turnip shaped in 3 days at temperature 22 degrees. When incubated at 37 degrees in liquid media such as 1% peptone water, it forms a fine surface pellicle which on shaking breaks up to membranous pieces, but the growth of Vibrio 0139 and Vibrio L-Tor and non-agglutination Vibrio cholerae has different. You can see on these pictures, on this growth of Vibrio cholerae serogroup of 139, this growth of Vibrio L-Tor into test tube with 1 peptones waters, and this growth of microorganism non-agglutination Vibrio cholerae. Transport nutrient media. For transport biological material from between different laboratory, you will use transport nutrient media, VR medium, Carey-Blair medium, autoclaving, seawater. For accumulation of Vibrio cholerae, pure cultural, you will use alkaline peptone water. Vibrio survives and replicate at high pH. Other organisms are killed or do not multiply. I told that microorganism Vibrio cholerae very well grows to pH 8.2. And for isolation and identification on the first step, investigation of biological material, we will use plating media, alkaline bile salt agar BCI, and important for us TCBS agar, the colonies of Vibrio cholerae, because Vibrio cholerae sucrose fermenting microorganism are yellow. And for cultivation of Vibrio cholerae, we will use Mansour's GTTA agar. After 22 hours incubation, Vibrio produce small 1 to 2 millimeter translucent colonies with grayish-black center. On surface agar, TCBS agar, you see yellow colony, Vibrio cholerae on TCBS agar and Vibrio parahemolyticus on TCBS agar. Colonies of Vibrio cholerae on TCBS agar are large, 2 or 4 millimeter in diameter, and yellow because of the fermentation of sucrose. They are characteristically round, smooth, glistening, and slightly flattened. This is medium of choice for the isolation of Vibrio cholerae and is widely used worldwide. TCBS agar is highly differential and selective nutrient media. But this nutrient media, TCBS agar, is not suitable for direct testing with Vibrio cholerae or 1 antisera for study about serogroups. For understanding, for identification of Vibrio cholerae, we will use string test. Vibrio colonies identified by string test, loop full of growth mixed with a drop of sodium dioxide on a slide. Positive test indicate by loss of turbidity, mucus appearance, formation of string as the loop is drawn away from suspension. You see positive reaction of string test. Biochemical reaction. Vibrio cholerae catalase and oxidase positive, fermentative producing acid but not gas, ferment glucose, maltose, mannose, and sucrose, do not ferment arabinose and lactose. Indole is produced, reduced nitrites are nitrates, methyl red and urise negative, Cholera red reaction we will use for identification of Vibrio cholera. Principle: few drops of concentrated sulfuric acid added to 24 hours peptone water culture produce red color. On this slide you can see test catalase positive, you see a bubble of gas, catalase negative Vibrio cholera, catalase positive, oxidase positive, you can see change color of indicate paper. This triplet sugar Kliegler agar, triplet sugar agar, microorganism Vibrio L-tor, for example, this ferment of lactose, do not ferment lactose, a ferment of glucose without gas, and this produce indole positive reaction and reduce nitrate to nitrates, this positive reaction, blue color. Differentiating Vibrios from related genera, we will be detection of Vibrio cholera between different genus, between Aeromonas, Pseudomonas, and Plesiomonas. Characteristic of Vibrio oxidase fermentation reaction, test Hewlett positive. Utilization of amino acid lysine and ornithine, but don't ferment arginine, and positive string test. Classification of Vibrio cholera. Vibrios were classified by Herberg, serological classification by Gardner and Wenkterman during reaction agglutination O1 and O139. Two biotypes of Vibrio cholera, classical and L-tor biotypes, and phage typing. I will explain what does it mean, this classification. For example, OF test. OF test give help for us understanding about OF test, oxidase fermentation test, and will be positive. You see a yellow color. Principle: this reaction, fermentative activity of Vibrio cholera, of glucose in aerobic and anaerobic conditionally. Oxidase fermentation reaction, Hugh-Leifson test, will be positive. Herberg's group of Vibrios classified by biochemical activity fermentose of carbohydrate, fermentation of mannose, saccharose, and arabinose. And Herberg groups includes 7 groups of Vibrio cholera. You see 7 groups, different type of fermentation, different saccharose, mannose, saccharose, and arabinose of Vibrio cholera. Antigenic classification of Vibrios by Gardner and Wenkterman. Vibrio includes 2 groups, Group A and Group B. Group A is cholera Vibrios, biochemical similarities, common H antigen Vibrio cholera. Group B, biochemically and antigenically heterogeneous. For us, interesting Group A. Group A subgroup, serogroups and serovars includes O1 and O1, non-O1 or O139 serogroups. O1 serogroups includes 2 biotype, classical and L-tor biotype, and group serogroup O1 includes serovars Ogawa, Inaba, and Gikoshima. What principal reaction, principal for classification by serovariants, serotypes of cholera Vibrios. This classification of different antigens or antigens during reaction of agglutination with antiserum O1, antiserum A, antiserum B, antiserum C. If you will see positive reaction with O1, A, and B antigen, it's Ogawa serotype. If you will see positive reaction with antiserum O1 and AC, you will understand about another serotypes of Vibrio cholera. If you will see positive reaction of agglutination with antiserum O1, A, B, and C, you will understand that Vibrio cholera serotype of Gikoshima. On this slide you can see positive reaction of agglutination with antiserum, this negative reaction with antiserum. Difference between classical cholera and L2 Vibrio. Test which help you for understanding about what biotype of cholera cause infectional disease by hemolysis properties, Voges-Proskauer reaction, check erythrocyte agglutination, polymyxin B sensitivity, group 4 FH susceptibility, L2 FH5 susceptibility. You see different properties between this test, between Vibrio cholera classic and L2 Vibrio. And next classification of Vibrio cholera, 5 steps of strength of classical biotype of Vibrio cholera O1, sensitivity to different phase groups, and classification phase type of strength of L2 biotype of Vibrio cholera O1, different group, sensitivity to phase different groups. What does it mean non-O1 139 Vibrios? These groups of Vibrio cholera includes serogroups O2, from O2 to O138. The first clinically cause gastroenteritis. Never cause epidemic cholera, extraintestinal manifestations like otitis media, wound infection, bacteremia, occupation or recreation, explosives, so water, seawater. For your understanding, during reaction of agglutination with antiserum O1, you will see negative reaction. Virulence factors of Vibrio cholera. Cholera toxin, if short CT, enterotoxin, endotoxin because it's gram-negative microorganism and contain lipopolysaccharide, adhesion factors. Adhesion to the epithelial surface and colonization may be facilitated by special fibria such as the toxin coagulated pilus, TCP. Gamma-glutamine protease formed early now as cholera lectin, cleavase, mucus, and fibronectin. Ferment of neuraminidase, and throughout the course of infection, the vibrios remain attached to the epithelium but do not damage or invade the cells. Mechanism of action of Vibrio cholera. Cholera toxin consists 1A and 5B subunits. The B bonding subunits of cholera toxin binds to the ganglioside GM1 receptors on the intestinal epithelial cells, which promotes entry of subunit A into the cell. The A active subunit, on being transported into the enterocyte, dissociated into two fragments, A1 and A2. The fragments A1 enter the cell and activates adenylate cyclase and increase cycle levels adenylate monophosphatase, cycle AMP. Subunit A2 bonding with B subunit and attach another intestinal cells. Cycle AMP cause activation of ion canals. Cycle AMP inhibits adsorption of sodium, activates secretion of chlorine, and water moves passively to gut lumen from epithelial cell to gut lumen. Loss of fluid and electrocytes cause shock and acidosis. On these pictures you can see mode of action cholera toxin. Epidemiology. 7 recorded pandemic. First pandemic in 1817 to 1823. Second pandemic in 1826, 1837. Third pandemic in 1846, 1863. Sixth pandemic in 1863 and 1879. Fifth pandemic in 1881, 1896. Sixth pandemic in 1899, 1923 years, and seven pandemic in 1961, 1991, characterized by causative agent of Vibrio cholera biotype L-tor. Began in Asia in 1961, L-tor quarantine disease, dysentery-like illness with gangrene of colon. On this slide you can see endemic region cholera. Cholera is an exclusively human disease. The source of the contamination is usually the feces of carriers or patient with cholera. Infection is generally spread by contaminated water or food such as uncooked seafood or vegetables. Mode of transmission: fecal oral. Clinical pictures: incubation period 1 to 4 days, acute onset, lightness, vomiting, profuse water diarrhea, rise water stool, rapid dehydration, 10 from 20 liters during days, hypovolemic shock, and cause death of your patient. Clinical cholera characterized of that cholera disease: no intestinal invasion, don't cause bacteremia, don't cause septicemia, and lesions of intestinal tract are pathophysiological. You can see pictures of sick individual of cholera, and these cholera hospitals, you see a special place for sick individual of cholera, because Vibrio cholera cause cholera disease, profuse diarrhea. You see special place for sick individual. Laboratory diagnosis: specimen, stool, rectal swab. For understanding about Vibrio cholera, you will use microscopic technique, dating motility, dark field microscopy. You will study cultural properties on surface: TCBS agar, yellow colony, alkaline peptone water. You will see different growth, type of growth, different species of Vibrio cholera. I saw you watched this slide. And you will use identification methods: light agglutination reaction with antiserum O1 and O139. You will use biochemical reaction for identification, group Heiberg's, if you remember, OF test, test of Hewlett-Sons. Study of differential properties between classical Vibrio cholera and Vibrio L-tor. I explained about different properties, and phase typing. Treatment of Vibrio cholera: rehydration, replacement fluids, electrolytes, and antibiotics, groups of tetracycline. Prevention control of infectious disease. My student includes two groups of prevention. It's non-specific method of non-specific prevention and specific method of prevention. Non-specific prevention: patient isolation, treatment, contact human, prophylaxis, antibiotic tetracycline group, pure supply of water, proper treatment, proper disposal sewage, and health education. Specific prophylactic or prevention, it's vaccine. We can use two types of vaccine. Very often we use oral vaccine, live oral vaccine, and can use kildural whole cell vaccine for people in high risk groups. Different country includes prophylaxis against cholera disease in a schedule of vaccination, and you will see, for example, a schedule of recommended immunization schedule, cholera for range of recommended ages for certain high risk group. What does it mean, high risk group? These people who have contact with sick individual, for example, doctors, who live in endemic region by cholera, who have a contact with cholera disease. These high risks of groups people. Today our lecture is finished. Thank you for your attention. Don't forget, take the computer test in your personal account. Thank you so much.